our next speaker is actually Dr. Schellenberg from the University of Pennsylvania in Philadelphia. And he's talking about genetics in PSP and CBD. And he's actually one of the leading authorities in genetics in the country. So please welcome Dr. Schellenberg.
Thank you for giving me this chance to talk to you a little bit about my research. I have no disclosures other than I get money from NIH and Cure PSP, private foundations, but no commercial firms. So, first of all, a lot of you people participate in drug studies as well as probably some of you in genetic studies. So I want to tell you why it's worth doing genetics. Why do I do human genetics? One of the advantages of human genetics is we're studying the disease of PSP, Alzheimer's, whatever, in the organism we want to treat, which is people. So there's no translation from animal models. Not that animal models are not useful, but you'd really like it to be right at the source. When we get a conclusion from the genetic study, we can directly apply it to understanding how the disease acts. So several reasons for doing genetic research. One is prediction. Right now, because there's no treatment for PSP, CBD, same for Alzheimer's, predicting somebody's going to get Alzheimer's is not greatly useful right now. But if we get to the point where we have preventive therapies, then you will want to be able to predict who is going to get the disease before they show symptoms. When you get a gene, you get a protein, you start understanding mechanisms, and you know what proteins are involved in the actual disease process. But the real driving force is, can we generate drug targets from our genetic studies? So going from a gene to a drug target, and not necessarily having to understand the mechanism completely. So I don't want to sell you a bill of goods. I want to say, you guys all should be saying, hey, does that actually work? Does it work? Can you prove to me it works? So other examples, there's one great example. In 2006, there's a gene called PCSK9, and people identified protective variants that loss of function. So normal people had lost half of the gene function, and those people did not get coronary artery disease. They're completely protected. So in 2006, there's this concept that if we reduced PCSK9, maybe we have a treatment for heart disease. Between 2006, a couple of companies that were paying attention developed monoclonal antibodies. They put them in a trial. 2011, 2016, they had an approved drug. Now, that is, to me, incredibly hopeful. Now, 10 years sounds like 10 years. That's a long time. So, but that is actually rapid for going from concept to approved drug. We hope it would be nice if we could do it more quickly, and I think there's hope for that. Since then, two other genes involved in cardiovascular risk were published. And in the same paper where they published this mutation, had this concept, they published preliminary clinical trials showing that it might actually work. So maybe even that 10 years can be shortened. This is a list of genes that there are drugs targeting, the genes where there's genetic information about these genes. Now, I don't want anybody to reveal personal health information. So typically I might ask, how many of you take statins? I take a statin, okay? So don't raise your hand. I don't want to know your personal medical history, but many people do. That's a genetically validated drug target, one of the most important preventative medications that we know of. Now, there's a gene MAPT. That is the gene that makes the protein tau that we've been talking about all day. And in 1997, there were mutations discovered in MAPT-tau in families that were autosomal dominant frontal temporal dementia, meaning 50% of the people descending from an affected person developed the disease. Very unusual families, and some of those mutations also cause PSP and CBD. It's interesting in that there's one that's cryptically named E10 plus 16. Mutation very dear to my heart. I've worked on it a lot. But most people with that mutation have a frontal temporal dementia, but a few people have PSP. We'd love to understand why, but we don't. when this happened in 1997, it really changed the way people looked at tau, because it showed that if you biochemically change tau, you would make tangles. So that just directly connected the tangle to the disease process, rather than just being junk that accumulated downstream as your brain degenerated. And a lot of the, it's also too, a lot of the mouse models use these mutations to effectively model the disease. For, as Dr. Clark said, that's incredibly valuable. Oops. Seem to be going backwards. So we have additional, those early studies focused on families. Now we can take just general populations of people with PSP and look most of the places. And we've come up with 4 genes, including MAPT. I won't talk about the top 3, but again, tau comes up, and these are common variances rather than rare. And the highest risk for having a MAPT, this common variant, is it increases your risk almost 6-fold. So Larry, or Dr. Goldby, was talking about the odds ratio of clusters of PSP patients of being 11. 6 is a pretty big number for a common risk factor. Now, that risk factor is H1. One of the things that's even shocked me as a geneticist is how messy your genome is. I always thought it was like, you know, you change one letter and all the other letters are the same. Our genome is a mess. There's all kinds of stuff going on. Fortunately, most of it doesn't make any difference. So anyway, so this is H1, H2. The H1 on the top is the risk form, and this whole billion base pair region is flipped around to give you H2. It's common. It increases your risk 6-fold for PSP. However, about 56% of the general population is H1, has 2 copies of H1, has the highest risk form. But your baseline, you start in 2 per 100,000, you multiply that by 6, you're still at 12 per 100,000. Still pretty rare. It's complicated. Here again, your genome is messy, so at one end of H1, all of these colored bars are different copies. Copy number changes that occur naturally, normally. We can go through and figure out that these are the 4 that actually make a difference in terms of risk. I think Robin made the comment at the scientific session yesterday, she rarely understands anything. Well, trust me, a lot of scientists do not understand everything that's presented. So here's a slide that you don't need to understand. But the same H1, H2 is also a risk factor for Alzheimer's disease, PSP, CBD, Parkinson's, and an interesting disease called ALS, Parkinson's Dementia Complex of Guam. So we're pulling out all of our tools, all the stops to genetic tools to understand PSP and CBD. We're using whole genome sequencing. So that way means if we can sequence all of a person's genome, there should not be any place where a genetic variation can hide. Now, in 2001, the draft of the human genome was released, came in under budget at $2 billion. So here's a question I want you to all answer. How many of, when you look in your bank account, see $2 billion? Because I'm going to hit you up for money if I see any hand, like, whoa. But I can't relate to that, but I bought a Honda Accord for $23,000 in 2001. If molecular geneticists had been in charge of Honda, that Accord would cost a nickel. Now, if we could do the same level of efficiency improvement. And, you know, I could take spare chains, I'll buy, you know, the month's worth of Hondas, I could give up my friends' Hondas, I could, you know, I wouldn't care about being towed. Anyway, so it's about $1,000 a genome now. 5 years, it'll be about $100. So it's a real shift in what we can do. We have funding from, we have funding from Cure PSP and the Tau Consortium and NIH to do 2,000 subjects with PSP. And this is more of your messy genome. There's deletions and insertions. They're incredibly common. And again, most of them you have to sort through and find the ones that actually make a difference. One thing that is, we have money to sequence people with PSP and CBD, but not normal controls. So we have to compare people with PSP to controls. Fortunately, the NIH, for a variety of different diseases, is sequencing, doing whole genome sequencing, and we can access that data to leverage our control. And this is just gobbledygook that is very important to me. Great, and as everybody, there's lots of people that contribute to this, and right now we work with samples from over 2,500 PSP subjects, and really appreciate that contribution, and 400 people with CBD, and thousands of controls that come from other projects, and so we can't do any of that without that level of participation. Thank you.